In the first video of this series, we introduced the topic of the origin of life and raised some questions that many people have assumed were answered. We promised that we'd address these questions in our follow-up videos, and here we are. As discussed at length in the fourth video, abiogenesis assumes that the first life form was a simple cellular organism, which could evolve by itself during the prebiotic era. And then, as time goes by, this simple cell starts to get more functions, becoming more complex, and forming more intricate life forms. As we are discussing the origin of life, we should be more concerned about that assumed simple cell. Just how simple could the cell actually be? And what is the minimum abstraction that can be assumed for a cell to be considered alive? That is, what are the minimum required biological components to have a living organism? Let's begin by taking a look at some of a cell's basic components. Dr. Nita Sahai is a professor and Ohio research scholar in the Department of Polymer Science and Polymer Engineering at the University of Akron. She gave a detailed lecture called The Origins of Life, From Geochemistry to Biochemistry, in 2014. In it, she described the basic minimum components of existing life, and these are heritable, mutable genes which are found in DNA, metabolic cycles which are proteins and enzymes, and a boundary membrane we call lipids. However, the relationships between DNA, proteins, enzymes, and the cell's membrane present a biological conundrum. You should be aware of the famous question, which came first, the chicken or the egg? A rhetorical question without a definitive answer. Mathematically speaking, the odds and probability of even one of these things arriving spontaneously generated and ready to function is infinitesimally small.
And may the odds be ever in your favor.
So, these basic components have a few chicken or egg kind of problems. Firstly, which came first, metabolism or replication? DNA or the DNA polymerase enzyme? This is a very complicated question with a lot of scientific study involved. Suffice it to say that this is an ongoing debate still far from being settled within the scientific community. Secondly, which came first, DNA-coded information, or cell machinery capable of reading this information to perform all cell functions? How could DNA contain information if it has no physical means of reading this information? And if the information cannot be read, it cannot be put to use. On the other hand, how could a machine be built? Wait, we can't say built, too close to design. Okay, considering that there's no real pressure for natural selection to begin, why would chemicals form into the shape of something that is used to read information, if there is no information to read? Thirdly, enzymes are necessary for the timely formation of proteins. However, these enzymes are partially built with proteins, so proteins cannot be formed without enzymes, but enzymes cannot exist without proteins. Lastly, proteins are required to create ribosome machines, but ribosome machines are required to create proteins. Back to mathematics. The idea of having all of these components arriving together, at the same time, in the same place, joining together perfectly, is extremely improbable. In addition, multiple other issues are still yet to be explained, like the prebiotic chemical formation of lipids, carbohydrates, proteins, and amino acids by natural processes like natural selection, time and necessity, and the origin of the information, the instructions, or the software encoding within DNA or RNA. That leads us to a concept proposed by famous biochemist Dr. Michael Behe called irreducible complexity, which refers to a system that has several components, which interact with each other, to produce a function that the components can't do by themselves. And, if you take one or more components away, the system doesn't work. So it is irreducible. That concept can be applied to very simple cells, to larger eukaryotes, to organs, and even to entire organisms. This idea can also be applied to machinery and equipment, as most devices are built from components. And, there are core components that can't be removed or else the device will not function. Sometimes the overall idea is used to counterargue Darwinian evolution, but we are focusing here on the origin of life, showing how the biology of primitive cells exhibits irreducible complexity. The simplest forms of life are single-cell bacteria. Viruses are simpler than the simplest known cell, but they lack the structures required to be considered alive. as they reproduce only when their genes take over the host cell's reproduction machinery. Nonetheless, even viruses require a minimum number of parts to function. It's also becoming clear that the living cell contains a lot of nanomachinery, and using the term machines automatically suggests the following: the composition of parts where some are irreducibly complex, functioning for a specific purpose, performing maintenance and correction, and, lastly, design. Scientists still have no proposal for what the simplest cell should look like, or how the least required components came together to start that assumed cell. The living cell looks like it was designed, functions to achieve a certain purpose like a good design, and contains parts working together in harmony as if expertly designed by an inspired engineer. Let's look at some specific examples. Bacteria flagellum. The following video clip is borrowed from a video called Amazing Flagellum. Michael Behe and the Revolution of Intelligent Design, published by Discovery Science on their YouTube channel.
In Darwin's Black Box in 1996, Behe spotlighted and made famous a number of really interesting discoveries that had been occurring in biochemistry and cell biology over the last 2 or 3 decades. At the level of individual cells, there are little tiny examples of nanotechnology, little tiny machines at work. The flagellar motor is the one that Behe made most famous. It's a rotary engine that powers a whip-like tail that functions like a propeller. And the machine in some bacterial systems turns at 100,000 RPMs in one direction and can reverse direction on a quarter of a turn and turn 100,000 RPM in the other direction.
Then we have microtubules. The following is a clip borrowed from a video called Microtubules, Tent Poles and Railroads, published by Phospho Biomedical Animation on their YouTube channel.
Microtubules are the tent poles that give stability and rigidity to your cells. They're made up of a protein called tubulin, which comes in 2 flavors, alpha and beta. Alpha and beta tubulin bind together in what we call a dimer, and that dimer binds to other dimers until you get a spiral that goes all the way around. So for a single microtubule, you easily need more than 1,000 individual tubulin proteins. If one microtubule needs thousands of proteins, that means that every cell must have many copies of both alpha and beta tubulin. Since alpha and beta tubulin only bind to one another in a very specific orientation, they automatically assemble in the right way to form a microtubule that can dynamically extend. Because they form a network that goes pretty much anywhere in a cell, and because they're generally quite sturdy, microtubules are the train tracks that you can transport cargo down. Cellular cargo needs a molecular freight train to carry it along the microtubules, and that is a protein called kinesin. Kinesin is able to bind to tubulin with its 2 feet-like motor domains and slowly work its way up the microtubule to deliver its protein cargo.
Our last example is DNA copying machines. The following is a clip borrowed from a video called Animations of Unseeable Biology, presented by Drew Barry and published by TED on their YouTube channel.
The astonishing molecular machines that create the living fabric of your body. What I'm about to show you is an accurate representation of the actual DNA replication machine that's occurring right now inside your body, at least 2002 biology. So DNA is entering the production line from the left-hand side, and it hits this collection, this miniature biochemical machines that are pulling apart the DNA strand and making an exact copy. So DNA comes in, it hits this blue donut-shaped structure, and it's ripped apart into its 2 strands. One strand can be copied directly, and it can be seen spooling off down to the bottom there. But things aren't so simple for the other strand because it must be copied backwards. So it's thrown out repeatedly in these loops and copied one section at a time, creating 2 new DNA molecules. Now, you have billions of this machine right now whirring the right way inside you, copying your DNA with exquisite fidelity. It's that mechanical. It's molecular clockwork. This is how you work at the molecular scale. So with a little bit of molecular eye candy, we've got kinesins, which are the orange ones. They're little molecular courier molecules walking one way. And here are the dynein. They're carrying that broadcasting system, and they've got their long legs so they can step around obstacles and so on. So again, this is all derived accurately from the science. The problem is we can't show it to you any other way.
There has been an outcry by some that supporters of intelligent design have coined the term machines to further their own agenda. However, the term machines, or something similar, has been widely used in multiple articles with impressively qualified authors. Let's take a quick look at those articles' titles and who their authors are. The Cell as a Collection of Protein Machines, Preparing the Next Generation of Molecular Biologists, written by Dr. Bruce Alberts. Polymerasis and the Replication, Machines Within Machines, co-authored by Professors Tanya A. Baker and Stephen P. Bell. Eukaryotic Transcription, an interlaced network of transcription factors and chromatin-modifying machines, written by Professor James T. Kaganaga. Mechanical Devices of Spliciosome, Motors, Clocks, Springs, and Things, co-authored by Professors Jonathan P. Staley and the late Christine Guthrie. Molecular Movement Inside the Translational Engine, co-authored by Professors Kevin S. Wilson and Harry F. Knoller. The HSB-70 and HSB-60 Chaperone Machines, co-authored by Professors Bernd Buchau and Arthur L. Horwich. Alright, let's wrap it up. It should be obvious to you, after this video, that a single-celled organism can't be called simple, as they have some basic parts that are complex enough for prominent, well-educated scientists to refer to them as machines. Using the information at hand, how can it be reasonably concluded that natural processes, namely natural selection, time, and necessity, were miraculously able to manipulate chemicals to form these complex structures? Is it really unreasonable to view these incredible constructs as being intelligently designed? In short, were these complex machines happy accidents of impossible odds, or were they designed intelligently? Thank you for watching. If you've enjoyed our video, don't forget to click the like and subscribe buttons below.